Rahim. This is the second video on the chapter on protein synthesis, and we're going to discuss the MCQs. Starting with question number 32, which we left off in the previous video. Diagram shows the stages of the production of polypeptide, DNA nucleotide sequence, template strand, mRNA sequence, and amino acid sequence. Which features of the triplet code is illustrated by the information given? An amino acid can be coded for by more than one triplet. Now you can see LEU and LEU, and they're two different triplets for that. Then question number 33. A peptide consists of 10 amino acids of 4 different kinds. So we have, say, amino acid number 5, and then we have 5 again, and then we have 7, and then we have 8, and then we have 9, and then we have 5, and then we have, so it consists of 10 amino, 1, 2, 3, 4, 5, 6, 8, and then again 9. And then 8, and then 9 again. So there are 4 different kinds. So it's only 5, 7, 8, and 9. And the total of 10 amino acids. What is the theoretical minimum number of different tRNA molecules required to translate the mRNA for this peptide? Minimum tRNA. So there will only be 4 tRNAs, because every time the tRNA, which will bring number 5, like this is a tRNA, and it brings number 5, so there will be 5, it will always bring 5, then it will again bring, so there will be 4 different types of tRNA. This is probably AAA, and then there's another one which is UUU, which always brings number 8. So it's only 4, 5, 8, 7, and 9. The same numbers are being repeated. So this is amino acid number 5, amino acid number 7. You see, I've just numbered the 20 different amino acids with the different R groups. So I've numbered them 4. So they're actually 10 amino acids, but there are only 4 different kinds. So there'll be 4 different types of tRNA molecules. Then coming on to question number 34. Which statements about tRNA structures are correct? There is a binding site for the attachment of a specific amino acid, as well as a different binding site for the attachment to the ribosome in order to allow translation to occur. There is a ribose phosphate backbone with a strong covalent phosphodiester bond in areas where the polynucleotides where base pairing by hydrogen bonding occurs. There is a section known as an anticodon that contains the same triplet of bases as the triplet of DNA bases that have been transcribed to produce the mRNA codon. So we have to understand is there is something which is wrong in any one of these statements. So the answers to this was 1 only, 2 and 3, 1 and 2, 1, 2 and 3. Now which one is correct is, of course, why is the answer B? Why is 3 wrong? There is a section known as an anticodon that contains the same triplet of bases as the triplet of DNA bases that had been transcribed to produce mRNA. So look at how this is wrong. If the DNA was TTT, then the mRNA is what? AAA. Then what is the tRNA? That's UUU. So this section known as the anticodon contains the same triplet of bases as the triplet of DNA bases. No, this UUU is the tRNA. This is the DNA is TTT. So that is why 3 was wrong, and the answer is 1 and 2 only. Then we come on to the genetic engineering. In a genetic engineering experiment, a piece of double-stranded DNA containing 12 into 10 raised to the power 3 nucleotides coding for a specific polypeptide is transcribed and translated. What is the total number of amino acids in the polypeptide? Now, this is a very, very favorite question which they keep on asking you a lot of times. You see, what we have to understand is that, say there are, why do we do the half of this? 12 into 10 raised to the power 3. So this would be, you have to see that if we have say 100 amino acids on this side, sorry, 100 nucleotide bases on this side, so we would 100 on this side. So this would be total 200. So this is A, T. It's saying DNA containing so many nucleotides. So this would be containing 200 nucleotides. Now you know that AAT or TA. Now this codes for one amino acid. So first you have to half this or divide it by 2, and then you divide it by 3. So it will be 6 into 10 raised to the power 3, and then you divide it by 3. Why do you divide it by 3? Because every 3 bases code for one amino acid. So 10 on 100 on this side, 100 on this side. So this was the total was 200. They gave you this figure 200. 12 into 10 raised to the power 3 was the number. So you know that half of these are on this side and half of these are on this side. So 6 into 10 raised to the power 3 on one side, 6, but then you have to divide it by 3 because every 3 bases A, T, G, C, A, T, these code for one amino acid. So that's why the answer was 2 into 10 raised to the power 3. And question 36. What is the correct sequence for the process involved in the formation of an enzyme in a cell? Transcription, translation, condensation, ionic bonding. Then the diagram shows part of a DNA molecule. Question 37. Which row correctly identifies the structures labeled 1, 2, 3, and 4? Now you see the 3 bonds here. So this has to be cytosine and guanine or vice versa. This has to be adenine or thymine, or this has to be thymine and this has to be adenine. Similarly, this can be guanine and cytosine. Now this, but they didn't ask you this. They said which row correctly identifies. So 3 had to be guanine or cytosine, right? And 4 had to be adenine or thymine because it's a DNA molecule, so that's not uracil. So adenine and thymine 4 was only this and this. Then you have to figure out which one was further. 1 was what? What is this 1? DNA. D for deoxyribose. So deoxyribose sugar was C. And 2 was phosphate. 2 was phosphate. So this was easy to figure it out. I don't think this was a very difficult one, but of course if you don't remember that C and G is 3 and A and T is 2. Question 38. Bacteria were grown in a medium containing N14 and 15. After several generations of all the DNA containing N15, some were transferred to a medium containing N14. Bacteria are allowed to divide once. The DNA of some of these bacteria was extracted and analyzed. The DNA was all hybrid DNA. So originally this, then we have, so original N15 is this, and then we have N14 was this. Some bacteria from the medium with N15 were transferred to medium N14. The bacteria were allowed to divide twice. The graph shows the percentage of N14 and N15 in the DNA of these bacteria. So you can see this is the, now you have N14 was percentage of DNA, N14 and N15. Now when would this be like? Now you have to make a further diagram to really, for me to explain this to you. As you can see in this diagram, now first it was only N15, then we have a hybrid, and then we have two hybrid, and then we have the two light ones, the N14 ones. So these are the 50% N14 and the N14 15, this is the hybrid one, is these. So 50%, this is this stage. Now it says some bacteria from the medium with N15 were transferred to medium N14. The bacteria were allowed to divide 3 times. What is the percentage of N14 and N15 the DNA extracted from this? Now, as you know, as I've I think just discussed this before also, the N14 and N15 would be only 25%, and the rest of this would be 75%. I'll just show you a diagram to explain this. Now please look at this diagram and just pause the video for a little while and then look at it how I've calculated it and why is the answer C. So the two purple ones are the hybrid ones, and this would be the 25% here. And the 75% is this one. Ones which are totally the light nitrogen, which is the N14. So please just pause this video, have a look at it, understand how DNA replication occurs, how we're going to get one heavy nitrogen and one light nitrogen, and how this is going to be. Then finally the answer is going to be C to this. Question 39, the table shows the tRNA anticodons for 4 amino acids. Asparagine, UUA, glutamic acid, and please remember it's tRNA. You don't read that carefully and then you make a mistake. Glutamic acid, CUU. Whenever there's U in it, you know it has got to be some RNA. Either it's mRNA or tRNA or ribosomal RNA. A cell makes a polypeptide with the following amino acid sequence: glutamic acid, asparagine, threonine, and proline. What was the sequence of bases on the DNA from which this was formed? So now they've given you a bit of a chore. They've said, okay, glutamic acid was the first one. So let's look at glutamic acid. This is the tRNA. So what will be the mRNA? And then calculate the DNA. So you have to understand is how do you do that? From CUU, the mRNA will be GAA. And then the DNA will be CTT. If you get confused, then you sort of write under it that if you get this wrong, if you're feeling you get it wrong, then you say GAA. So this would be the DNA, this would be the mRNA, and let's calculate the tRNA then. So the tRNA will be CUU, but you have to remember in the RNAs you've got to have U's, and in the DNA you've got to have the T's. If you can't do that, you can do it in another way, which I ask you to do that, is that you do TTT. If this is the DNA, then the mRNA would be AAA. So DNA is TTT, mRNA is AAA, then the tRNA now has to be, if you're going to do it the DNA method then, but then you need to convert these into U's, because RNA only has U's, doesn't have T. So even if you've got a T written, you can do it the DNA method, then convert the T's into U's. Whatever suits you, you've got to figure that out. Then the next one was asparagine. Now asparagine is this here, and the was UUU. So then you've got to figure out how you're going to change the UUA. UUA is asparagine. So this is the tRNA. Always write it because you confuse this all the time. Then convert it to mRNA and then convert it into DNA. So AAU and then TTA. Then you figure out where is TTA. So this is CTT and TTA. Well, this is the same here, same here. So the third one will decide. And the third one was threonine. Now threonine is this. So UGG converted to this is tRNA, so mRNA, ACC, and then the DNA will be TGG. So here was the one that you were going to figure this out. TGG. I might not have written it rightly. Okay, so this is one way that you solve these sort of questions. They're a little tough, but if you got the hang of it, it's easy. What does the enzyme RNA polymerase synthesize? mRNA from a DNA template. RNA polymerase would have to make some sort of an RNA, and the mRNA comes from a DNA template. The diagram shows part of a DNA molecule. How many hydrogen bonds are involved in holding these strands together? Now that was very basic. I've talked about this number of times. AT2, GC is 3. So GC is 3, 3, 3, and 2. So 11. Then question number 42. If you're struck at it, please look at the exam report of this. The year and everything is written on the top of every question, so you can easily look through the exam report. I've just been asked a question on the YouTube, and all that student needs to do is find out the exam report and read up on that question if he or she is not clear. The table shows the percentage of bases DNA from various types of cells. So you get all these percentage of bases: calf thymus, bull spleen, bull sperm, rat bone marrow, and yeast. What is a valid deduction from these data? Now there's a way you do this question. You say, does the data tell me this? So every time you read one answer, you say, does the data tell me all cells from the same species have approximately the same content of DNA? Does the data tell me small differences of DNA differences in DNA from different cells have large effects? No, you can't even see the effects of it. It's just telling you the percentage of bases of the DNA. The 4 bases show complementary base pairing. Well, does it show me that? 28.2 adenine, 27.8 nearly the same as thymine, 21.5 guanine, so 21.5. 22.5 cytosine. Does it show some sort of a correlation? Yes. 27.9 adenine, 27.3 thymine, then 22.7 guanine, then 22.1 cytosine. So that's the only thing. Then it says, how is D incorrect? The structure of DNA is different in yeast and animal cells. No, this data does not tell me. So every time you say read the answer, just ask yourself, does the data tell me this? Does the data give me this information? Does the data give me this information? And read A, B, C, and D, and then see if you can figure this out. Then coming on to question number 43. Which row shows the correct combination? Triplet code DNA, codon, mRNA, anticodon, tRNA. Yeah, triplet code DNA translates and codon is on the mRNA and tRNA is on the anticodon. So I always say T and T here. And mRNA, I say MC. mRNA has codon, medical certificate. Coming again to this question, which is again a Matheson star experiment. You need to look up this Matheson star experiment. That will make life easy for you and understand it. There are many YouTube videos on that. A culture of bacteria had all its DNA labeled with a heavy isotope of nitrogen N15. A sample was taken and spun in a centrifuge. The diagram shows the position of the DNA band at Z in the centrifuge tube. The culture was then allowed to reproduce using nucleotides containing the normal isotope of N14. Sample was taken and spun in a centrifuge after 1 generation and again after 2 generations. In which pattern would the DNA be found after the first and after the second generation? In the first one, they would all will be at Y because they'll be heavy and then there'll be a light nitrogen. You see, if you look at the example that I gave you, first all heavy, then one heavy and one light. And I'm just giving it a squiggly line to show you the light one. So that would all be here because it's all hybrid DNA. It's neither heavy nor light, it's just medium DNA. And then what would be after the second generation? We would have this and then we'd have this, then we'd have this, and then we'd have this, which I've shown you before in another question. So half at x, so all the hybrid would be here, the hybrid would be here, and the light would be up, which is this one, and the hybrid is this one, which is this one. So half at x and half at y. Question 45. DNA said to replicate in a semi-conservative way. Results of Matheson and Stahl experiment gave overwhelming support to this theory. They used E. coli, which has a generation time of 50 minutes. Here are the steps in the experiment. They are in the wrong order. All bacteria contain 15 N DNA. All bacteria contain hybrid DNA. Bacteria contain either all N14 DNA or hybrid DNA. Bacteria grown in N15 medium for many generations. Bacteria transferred to N14 medium and sampled every 50 minutes. So S was the first one. Then P, all bacteria contain 15 DNA, so S and P. And then T, bacteria transferred to N14 and sampled every, so S, P, T. And then Q, all bacteria contain hybrid DNA. And then R, bacteria contain either all N14 or hybrid DNA. So this was the sequence. Please read this again, understand it, and then comprehend it. Last question 46. What would be the result of analyzing part of a DNA molecule? So now you read all of them very carefully. Hexose sugars and phosphates in equal proportion and an equal number of cytosine and guanine bases. No, how can there be an equal number of cytosine and guanine bases? There's adenine, thymine, there could be 10 ATs, 5 GCs. There could be 80, 80, 80, 80 3 times. Then there could be maybe 10 times GC. Nucleotide and phosphates in equal proportion and an equal number of adenine and cytosine bases. Pentose sugars and phosphates in equal proportion and an equal number of adenine and thymine bases. Yeah, naturally, if there are 3 adenines, then there are going to be 3 thymines. If there are 2 guanines, there are going to be 2 cytosines. And then the D part says twice as many phosphates as pentose sugars and an equal number of adenine and guanine. Adenine and guanine do not pair up with each other. And twice as many phosphates. So you've got to understand what is the structure of a nucleotide. This is the pentose sugar, 5 carbon sugar. This is called deoxyribose. And then there could be adenine or any other of the bases, adenine, thymine, guanine, and cytosine. And then there is a phosphate attached to it. This is the structure of a nucleotide. So how can they be, if there are 10 of these, then there have to be 10 of these. The Y is saying twice as many phosphates as pentose sugars. That's absolute rubbish. So if there are 10 deoxyribose sugars, then there are 10 phosphates. If there are 10 adenines, then there will be 10 thymines. If there are 10 adenines, then there will be 10 thymines. There can't be 10 guanines. Guanine does not pair with the adenine. So I think that should be a pretty good exercise for you to do these MCQs and comprehend this chapter in a better manner. Thank you very much. Thank you for subscribing and thank you for watching the videos. And please do leave any comments and I'll try to reply as soon as I can. Thank you very much.